Hello everyone, myself Devika, first MSc Biology. Today I am going to discuss about the topic antigenicity and immunogenicity. Antigenicity. What is antigenicity? It is the property that allows a substance to combine specifically with antibody whether or not they are immunogenic. Explaining antigenicity with an example. Bacteria, which are non-pathogenic, enter to our body with the same epitope, and antibodies specific to this bacteria are already present inside the body. These antibodies bind to the invaded bacteria. Here bacteria did not induce any immune response. It just bind to the product of an immune response, that is the antibody. Moving to next term, immunogen. A immunogen is a substance that induces detectable immune response, that is a cellular or humoral response is called an immunogen. An immunogen can trigger an immune response and act as an antigen in that immune response. Thus all immunogens are antigens, but all antigens are not immunogens. Next topic, immunogenicity. Immunogenicity refers to the ability to induce humoral and cell-mediated immune response. Explaining with example, when an antigen enters to our body, they are recognized by plasma cells, and this antibody specific to this antigen are produced and bind to this antigen. Immune responses are produced. Factors affecting immunogenicity. There are four factors affecting immunogenicity. They are foreignness, molecular size, chemical composition and complexity, and susceptibility to antigen processing and presentation. Detailing the factors. First one is the foreignness. In order to elicit an immune response, a molecule must be recognized as non-self by the biological system. The capacity to recognize non-self is accompanied by tolerance of self, a specific unresponsiveness to self-antigen. Next point, much of the ability to tolerate self-antigen arises during lymphocyte development, during which immature lymphocytes are exposed to self-components. Example, the substance bovine serum albumin BSA is found in cow. BSA is collected from one cow and injected to another cow. The BSA is not immunogenic because BSA is already present within the body of cow. But when it is injected to a rabbit, it is strongly immunogenic because BSA is not produced in rabbit. Here it is a foreign to rabbit. It's all about the first factor, foreignness. The second factor is the molecular size. There is a correlation between the size of the macromolecule and its immunogenicity. The more active antigens have the molecular weight of 1 lakh Dalton. Substances with less than 5000 to 10,000 Dalton are poor immunogens, but mass less than 1000 Dalton have proven to be immunogenic. The third factor is the chemical composition and complexity. First point, synthetic homopolymers tend to lack immunogenicity regardless of their size. Next, copolymers composed of different amino acid and sugar are more immunogenic than homopolymers. Chemical complexity contribute to immunogenicity, and also structural complexity is also contributed. It is the primary, secondary, tertiary and the quaternary structures. Proteins are powerful immunogens. Carbohydrates act as immunogen if it has a complex structure or it is bound with a protein molecule. Lipids and nucleic acid are not immunogenic. When it is conjugated with a protein carrier, they become immunogenic. Next factor, susceptibility to antigen processing and presentation. For the development of both humoral and cell-mediated immune response requires interaction of T cell with antigen that has been processed and presented together with the MHC molecule. Next point, large insoluble molecules are generally more immunogenic than small soluble ones because they can be readily phagocytized and processed. Next, macromolecules that cannot be degraded and presented with MHC molecule are poor immunogens. Therefore, a substance must be degradable in order to be immunogenic. Also, highly stable and non-degradable substances are not immunogenic. And this is all about my topic. Thank you.